In this video, we're going to talk about the role of DNA. And before we begin, I just want to, kind of just break it down. This is your dad, this is your mom. You kind of had a little fun session and they made you, hopefully not accidentally. Never mind. Shouldn't have said that. Well, DNA is when we have information being passed on from one generation to the next. It's just that DNA encodes and it allows the transmission of genetic information from the parents to the offspring. And let's say that I was making this video before in 1949. I'll be telling you that, hey, dude, guess what? I'll be probably saying, what is the role of protein? And I'll be like, hey, look, your mom and dad, they made you through protein. And genetic information was passed through protein. That made no sense. And you might be asking yourself, why did they think that way? And the reason they thought that way really was because proteins had 20 different amino acids in comparison to DNA only has 4 different nucleotides. So they were like, hey, look, you have 20 different amino acids. They can probably do more than 4 different. They probably encode this genetic information compared to the 4 nucleotides. And we've discussed this before, that you have nucleotides that have the backbone structure of a phosphate and a sugar, and a phosphate and a sugar, and the only thing that's changing is the aromatic ring structure, which is the adenine, guanine, thymine, cytosine. These are the only 4 different variable parts. So it makes sense to think that, hey, look, you know what, the proteins might be passing on the information. And then we had somebody called Mr. Oswald Avery, and let me just draw a mustache for Mr. Oswald Avery because he's a very smart man indeed, very smart man. Maybe a little bit here as well, because he has to be famous for the chicks or the ladies as well. Because come on, it's Mr. Oswald Avery, guys. Come on. So it's 1944, and he says, "Hey guys, you guys are stupid. Y'all are stupid. Because guess what? What? Guess what? What? DNA is the active agent that allows the transformation of genetic information, and he proved it through an experiment, through the E. coli T2 bacteriophage experiment. And this is discussed in a future video. It's a whole different topic, but basically it's a viral infection that proved that DNA contains genetic information. And there's a lot about viruses on this page. And I don't like viruses, trust me. Coronavirus, not my thing. It's not your thing either, man, trust me. So here is the overall general central dogma of replication. Here's a central dogma, right? So DNA goes to RNA. I don't like this. DNA goes to RNA, and RNA goes to protein. This is the general sense of production inside our body. So DNA is preserved. It has to be translated. And I say translated. I'm sorry, I said that wrong. DNA has to be transcribed to RNA. Notice when I say transcribed, I'm saying there's a word scribed inside it. It's being copied. So DNA is copied into RNA, and RNA is translated. It's like you're translating it to a different language. It's translated into protein. And here is essentially what's happening. So this is our DNA, right? So first of all, we have a couple enzymes that come together. They kind of form, they unbind the double helix. And then we have some proteins that come together, and this is a very overview of transcription. Transcription is a process when we have, like I said, we have this double strand right here, and then it's unbinded, so proteins can come and synthesize a complementary copy. So let's say that this copy that's being made is going to be exactly the same. Is going to be exactly the same as this right over here. This strand over here is complementary to this, and this right here is exactly the same as this. But remember that mRNA does not have thymine. It has uracil. That's an important thing to remember. So, and then this mRNA can go outside the nucleus, and then it's translated. It goes from the 5' direction to 3' direction. And it's translating to protein. It's using all this instructions given by the DNA, and it's given by the DNA, and then these proteins are made. And it's important because, look, these mRNAs can be can be destroyed later on. But if we have DNA just going in, we can't destroy DNA. It's creating a lot of mess. And that's why we have mRNAs. And remember, something very important that I will be stressing throughout this whole time is that translation occurs in the 5' to 3' direction always. You will never see transcription or translation occurring from 3' to the 5'. That never happens. Please, transcription and translation is always happening in the 5', the 3' direction. Please remember that. And the way I remember is that when I say 5 and 3, I'm like, all right, you know what? The numbers got to decrease. It has to go from 5, 4, 3 to 1. So 5 to 3. Okay, got it. That's how I memorize it. Here's an exception to the central dogma of the central dogma was this theory right here. DNA goes to RNA, then RNA goes to protein. And the exception to the central dogma is the virus, because viruses can go from RNA to DNA. It's called reverse transcriptionase. What? Well, I can't say it. Well, I'm going to talk about that in later on videos. Transcriptase. I said it. Okay, good. So RNA can go to DNA, and that's a violation of the central dogma. And the second violation is the final products of RNA sometimes are not protein. Sometimes we have DNA being transcribed to RNA, and this RNA can be non-coding, or it could be just a functional RNA, which means it has other functions than protein. So here are some exceptions to the central dogma. Oh, okay, so we're going to talk about something called ambiguity. Ambiguity. Why can't I say words right now? It's 12 a.m., guys. I'm just making this video right now. Forgive me. So we have 64 codons. So let me just really quickly make you understand what codons are. So we have three different, okay, I want you to think about this, all right? So we have genetic code is written into 3 nucleotides, all right? So a nucleotide is adenine or thymine. So when we have 3 nucleotides, it's called one codon. And a single codon codes for a single amino acid. And this is proven through various genetic experiments, that we have 3 nucleotides that code for an amino acid. So generally, the first 2 nucleotides of a codon determines the amino acid, what amino acid is synthesized. So we notice that let's say that we have guanine, right? So notice this. We have guanine, right? Guanine. And then look at this. U, C, A, and G are coding for the same amino acid. And then if we have a mutation of the last nucleotide, sometimes it really doesn't matter that much, because it might have some structural changes to the entire structure of the amino acid, or the secondary structure or the quaternary structure. It might have a little difference on that, but really it's coding for the same thing. So it's mostly it's really not an issue. But if a mutation of the last nucleotide is not harmful if it codes for the same protein, and we just said that. And if it doesn't code for the same protein, and this protein has different properties and characteristics than the original one, let's say it's not hydrophobic compared to the original one, it's going to have major devastating impacts. I'm going to talk about that in later videos. So the degenerate code, more than one codon. Okay, so this is very interesting, right? So we have 64 codons in our body, right? That code for specific amino acids. So we have GAG, so GAG, valine. GAG is always going to code for valine. There's no ambiguity over here. I can't say this word again. Ambiguity. There you go. There is no confusion that GAG is gonna create valine. But valine is not just made from GUG. Did I say that before? Let me say that again. So GUG, right, is making valine. There's no debate. GUG is making valine, but valine can be made from different codons as well. Let's say we have GUU. That's also going to make valine. So that means that a codon is specific to an amino acid, but an amino acid is not specific to a codon. That's very important to understand. So there are 64 codons, and there are 61 coding codons in our body. There are 3 codons that don't really code for anything. They're just stop codons, and they're called nonsense codons. Here's degenerate code. A degenerate code is when you have more than one codon for the same amino acid. It's just a term, just memorize it. And then we have DNA methylation. CH3 is added, and this changes gene expression. DNA methylation is very interesting because this allows genes to be either silenced or expressed sometimes, and this is discussed more in future videos. So as you leave this video is giving you a highlight of everything that we are going to get in super detailed into in the future videos. This right here was a very overall rundown of the central dogma. DNA to RNA to protein. If you're watching this video for the first time, just remember that DNA goes to RNA to transcription. Transcription is just copying. RNA to protein through translation. Translation is when you translate something into something else from a message to an actual protein. That's all. And also this right here will be given to you, and you should get more familiar to this later on.